Moving on to the normal flora or commensal bacteria section. This is one of the sections as well that I get really excited with because this allows us to really individualize treatment plans for patients, as well as practice precision medicine, choosing the right product for the right patient at the right time given their lab tests that come back. We know that there is trillion of microorganisms that inhabit the human intestine that make up this complex ecosystem that plays such an important role in human health. Not only does it play a role in our immune system, not only does it play a role in the creation, absorption, and breaking down of nutrients, it also plays a role in our risk of diabetes, our risk of autoimmune disease, how our body is going to function when we consume the foods that we consume really impacts and is influenced by the commensal bacteria. Commensal bacteria extract nutrients from the energy from our diets. If they're imbalanced, you'll actually see in the next section when we go into the Firmicutes bacteriocytes ratio, we'll see if that ratio is off, you're more likely to extract the calories from the foods you eat, more inflammation, more risk of insulin resistance, prediabetes, etc. This normal commensal bacteria plays an important role in maintaining the gut function, produces vitamins, and protects against colonization of the gut by potential pathogens. The first normal bacteria we're going to talk about is Bacteroides Fragilis. This is a gram-negative species that falls into the Bacteroides phylum. You'll see that gram-negative species fall into Bacteroides, whereas gram-positive fall into Firmicutes. And this is just a way to classify different normal bacteria that's in the gut, the same way that we have animals. Animals can be thought of as normal gut bacteria, and then within animals we have dog and cat. And then there are so many different species of dog, so many different species of cat. That's how you can really think of this Firmicutes Bacteroides ratio. It's essentially the dog and cat classification, then all the species fall in. Now, when we're talking about gram-negative or gram-positive species, this just has to do with whether or not when we gram stain them, do they produce a fluorescent purple after we stain them and put them under a microscope. And this has to do with the cell wall structure. So again, Bacteroides fragilis is a gram-negative species. It is immune modulating. It is part of the normal gut flora, and it's believed to be involved in microbial balance, maintaining the barrier, and neuroimmune health. High levels may result from reduced digestive capacity, so we want to look at the intestinal health markers section and look at what the elastase is to determine if that is true. It could also result for constipation, so we want to say, okay, how can we improve motility in the gut, whether that's digestive enzymes or a higher fiber diet, more water, more dark leafy greens, lifestyle factors like that, chewing your foods, eating in that digest and rest phase. Low levels may contribute to a reduced anti-inflammatory activity in the intestines. So we want to look at calprotection. What is the inflammation like that's presenting in this patient in the intestines? Bifidobacterium is gram-positive, so it falls into the Firmicutes. It's present in breast milk. That's why we see higher levels more common in children than adults. And it also colonizes the GI tract at birth. Common in probiotics, and thrives on a wide variety of prebiotic fibers. So most oftentimes we're seeing bifido low. You definitely can support with a bifido-containing probiotic, but also looking at the prebiotic fibers to help fuel the development of this healthy commensal normal bacteria. You may also see an increased association between those who weren't breastfed or those who were born through C-section and having a lower level of bifidobacterium. Enterococcus is a gram-positive, lactate-producing. That's one of the short-chain fatty acids. Short-chain fatty acids are produced by these normal gut flora, and that helps to provide fuel sources for the intestinal cells. So the intestine provides the space for the bacteria to live, and the bacteria breaks down different nutrients from the foods that we eat and provides fuel source for the intestinal cells. So it's more of a symbiotic relationship that we're seeing. High levels may be due to reduced digestive capacity, constipation, potentially SIBO, or small intestinal bacterial overgrowth. So you want to look at what is the digestive capacity, so the elastase, and ask the questions about bowel habits. Low levels may indicate insufficient bacterial flora. Escherichia species, so this is the gram-negative species of Escherichia. It's part of the normal gut flora. This is not the pathogenic type of E. coli that we discussed in the previous sections. This is the primary species that's in the genus is E. coli, and most E. colis are non-pathogenic, right? So we have a lot of this non-pathogenic E. coli, which is actually protective against the pathogenic E. coli. So look at the first page to see if the pathogenic was there. High levels may indicate increased intestinal anti-inflammatory activity. So if you have an infection of some sort, what will happen is the Escherichia E. coli will increase because they're going to outnumber, but they're going to increase that inflammatory response. So if you have high levels of this could be a normal physiological reaction to the body trying to protect itself because of a pathogen is present. Low levels may indicate reduced mucosal health and decreased protection against the pathogenic E. coli. So something to consider as well. Lactobacillus, this one that is commonly found in probiotics. It's a bacteria in the Firmicutes phylum. High levels may result from reduced digestive capacity, so considering digestive enzymes or protein HCl, looking at the elastase marker, or excessive intake of carbohydrates, so seeing what their diet is like. In addition, the low levels could indicate that they just have a low carbohydrate diet or a high salt diet, or it could indicate mucosal health. So a history of the patient to understand what their dietary habits are. Are they on keto? Are they on standard North American diet? Could contribute to this lactobacillus growth or lack of. Clostridium falls into the gram-positive genus in the Firmicutes phylum. The Clostridium genus is diverse and consists of both pathogens and normal commensals, so both the beneficial and the potentially harmful, the same thing that we saw with the E. coli. So this is not the same as the pathogenic Clostridium difficile, those toxins that we saw in the first section. This is part of the beneficial commensal bacterium in the gut. High levels may, again, reduce digestive capacity or constipation. So you're seeing a trend here that you can think in the back of your head that if normal bacteria is elevating, we want to look at what is the digestive capacity? Is there constipation? What is their diet like? Is it high in carbohydrates? So keep that in mind. And low levels may be due to insufficient fiber intake. So really promoting that whole foods, lots of fibers, lots of whole grains, looking at adding in more fiber from those root vegetables, dark leafy greens as much as possible to promote the growth of the healthy normal bacteria. Enterobacter is the gram-negative bacterium that falls into the Bacteroides family, closely related to E. coli, but not the same. And again, we're seeing the same trends, high levels indicating increased intestinal inflammatory activity, and low levels may indicate reduced mucosal health. The next two are markers that were recently added to the GI map test, which I'm really excited about. The first is Akkermansia. And this one you can think of as the mucus degrader. So it degrades the mucus-derived sugars, and the metabolic products produced from that process support the growth and energy needs of other gut microbiomes. So it loves mucus. So what it does is it takes the sugars, breaks down the mucus, and then those byproducts of the breakdown provides the fuel and energy source for the other gut microbiomes or microbes to grow. When Akkermansia degrades the mucus, what happens is the goblet cells of the intestine are stimulated to secrete more mucus. This allows for healthy turnover of the mucus layer. You don't want that mucus layer to be very thin, because what happens is if that mucus layer is thin, that's your first line of defense with your immunoglobulins to prevent different pathogens from binding. Low levels may be associated with obesity and metabolic dysfunction, and high levels have actually been linked to multiple sclerosis. So I'm wondering if I could zoom this in here for you to see. Again, our Akkermansia here is really helping to sustain this mucus membrane. Yes, it degrades the mucus membrane, but through the degradation process, it's going to stimulate these goblet cells to produce more mucus, so there's healthy turnover. If you have lower levels of Akkermansia, we're seeing obesity, diabetes, inflammation, possibly due to too much alcohol intake, the mucus levels decrease. There is increased LPS toxins from the different pathogens that are able to bind. And then we're seeing things that seemingly don't relate to the GI system, such as liver dysfunction, insulin resistance, inflammation, type 2 diabetes. This all has to do and comes back to the gut health. Remember, the gut is the foundation. Really, really important to draw these links to practitioners so that they can see the value in measuring the gut microbiome, because it's implemented not just in gastrointestinal disorders, but a wide variety of other symptoms and chronic conditions that patients are coming in with. Okay, Fecal Transplants. This one is always challenging to say. It is widely recognized as a keynote species in the Clostridia class, as well as a major butyrate producer. And the butyrate, again, is the fuel for the intestinal cells and promotes an anti-inflammatory process. One of the questions I often get with the GI map test is why it doesn't measure short chain fatty acid distribution. So on some of the older technologies, the previous lab tests that were put out there, they would list percentage of different short chain fatty acids in the gut. And that was because they couldn't measure directly the bacterium that was producing it. So it was a roundabout way to get an idea of indirectly saying which bacteria was low. We don't have to do that anymore because we have the technology to identify which organisms are producing the butyrate. And as long as you know this is a butyrate producer, you can infer the client doesn't need butyrate. It needs diet, lifestyle, and supplementary advice in order to promote the healthy growth of the normal bacterium. In addition, in order to determine normal levels of short chain fatty acids that would be optimal, it is very challenging and often inaccurate. This is because diet dramatically influences the distribution of short chain fatty acids. In addition, basal metabolic rate, or BMI, dramatically influences the distribution of these short chain fatty acids. So to establish an accurate reference range would be very challenging based off of the samples that you're getting. Not everyone is controlled for their BMI and their diet. So measuring these percentage of short chain fatty acids, it's not accurate. It's a roundabout way to determine which commensal bacteria might be low or high, and we have a better technology to just measure those bacteria. So this is a way better indicator of what the short chain fatty acid production is like in the gut. Low levels may be associated with a wide variety of chronic inflammation or inflammatory diseases, as well as autoimmune diseases. The consumption of a higher quantity of animal meat, animal fat, sugar, processed foods, low fiber diet, so thinking the standard North American diet, the SAD diet, will actually reduce the amount of this bacterium. While a high fiber, fruits and vegetables, low meat, or at least moderate meat, grass-fed as much as possible, will help to enhance the number of the bacterium. So this is a diagram as well that's showing animal studies of a healthy individual. What this study is doing, it's actually a recent new study, is they're looking at optimizing an animal's GI tract with Protsky and transferring it into a healthy individual, specifically a diabetic patient. So this is fecal transplants from animals, looking at how we can positively impact diabetes and metabolic changes by influencing the gut. So I think it's a lot easier to do dietary, lifestyle, and supplement changes versus transplanting feces from mice. But this was really cool to see that this is the new research that is coming out there. So again, in summary, in general, a high carb diet results in overgrowth. Inflammation can be the root cause of overgrowth, as well as decreased digestive capacity and constipation. Low levels of these commensal bacteria can indicate reduced mucosal health, insufficient fiber, or a low carbohydrate diet. In terms of treatment of low commensal bacteria, you can consider things like probiome at 50 or 100, depending on the findings. Where I'm saying depending on the findings is you might want to look for something that has higher levels of straight Bifidobacterium if Bifidobacterium is the only one that's low. Increased dietary intakes of fiber, fruits and vegetables, that's going to help with short chain fatty acid production. That's going to help promote the healthy growth of the normal bacteria so they can make those short chain fatty acids to in turn support the energy needs of the intestinal health. Remove dietary sugar, remove refined carbohydrate, consider a prebiotic supplement, fermented foods, reduce inflammation, as well as address other imbalances that come up on the GI map test. Treatment of high commensal bacteria would really depend on looking at the additional findings on the GI map and treating accordingly. What is their digestive capacity? So looking at their elastase, do we need a digestive enzyme or betaine HCl? Remove dietary sugars, remove the refined carbohydrates, and we might consider treating the overgrowth if it is significant with a broad spectrum antimicrobial, such as GI Microbex and oil of oregano.